just don't get this.
Cells! Why? Professor?
You rang?
I just don't understand these cells.
Huh. Allow me to explain.
Leaders can do, we can too.
Professor Josh, is this a cell?
I believe so, Dave. I think it's a plant cell.
Plant cell. Don't they manufacture their own organic compounds for use?
Yeah!
So, if a plant manufactures its own food, how the heck does it do it?
Well, Dave, let me show you.
You know, this cell's pretty big. Is there a specific place where photosynthesis occurs?
Yeah, I think it's in those little green things over there. I think they're called chloroplasts.
So this is the chloroplast.
Here it is, in all its green glory.
Very green.
Right? You see, young David, before the plant cell manufactures organic compounds in the Calvin cycle, it first needs to create energy through the light reactions, which are located in the scenic thylakoid membrane. Right this way. We now bring you to the thylakoid membrane of the chloroplast, where all the action and fun of the light reactions of photosynthesis occur. To start off, light energy from the sun, that big yellow thing in the sky, hits pigments within photosystem 2, which is a collection of proteins and pigment molecules. To collect light, energy from the light is passed from electron to electron within the pigments until it reaches a specific wavelength, in this case 680 nanometers, which can excite an electron in the P680 chlorophyll A pair molecules within the reaction center complex of the photosystem. The P680 molecules pass the electron to a primary electron acceptor, which then passes the electron to the electron transport chain, similar to those found in respiration. The electron passes through a series of proteins within the electron transport chain, which generates a proton gradient, and through the process of chemiosmosis produces ATP. Woo! The electron then replaces one that was lost in the PS1, or photosystem 1 complex, and then again, energy from light passes through pigments till it reaches the P700 chlorophyll A molecule pair, similar to the P680 from photosystem 2. The electron is then excited up to the primary electron acceptor of PS1, which again passes the electron down an electron transport chain. However, this time, the energy from the electron is not used to drive ATP synthase, but instead is used to reduce NADP plus to NADPH. Woo! The electron pushed up from P680 back in photosystem 2 is replaced by electrons from a molecule of water, which is split from H2O to 2 protons, 2 electrons, and 1 oxygen. Electrons from the split water enter PS2 and replace the electrons lost from the P680 molecule. And the process begins again. How was all that, Dave?
Pretty good, I think I get it now. But, okay, so it makes some ATP and it makes some NADPH. But how does that make sugar?
Well, Dave, we need to take the energy produced within the NADPH and ATP molecules and follow it all the way to the Calvin cycle.
So, Professor, if that's a light reactions, that just produces ATP and NADPH. How do they make sugars?
You see, David, the ATP and the NADPH produced by the light reactions moves over to the Calvin cycle.
So, Professor, where does this Calvin cycle, as you say, take place?
All around you, David. Here in the stroma of the chloroplast. Here, where CO2 molecules diffuse into the organelle, where they are bound by the enzyme rubisco to the 5-carbon compound RuBP, forming a 6-carbon compound, which is attacked by ATP and phosphorylated, where it breaks apart into 2 3-carbon compounds. NADPH, also from the light reactions, reduces the compound into the well-known simple sugar 3G.
G3P.
G3P. Oh, it's the first time. G3P.
I think I get it. So this 3-carbon sugar, the G3P, that's what they make glucose out of? Wait a minute. You said Calvin's cycle. If the G3P is made and used as glucose, how is it a cycle? How does it go around?
Well, my banana-eating friend, ATP is used by the organelle to fix the remaining 3 GP molecule back to the original 5-carbon Ruby P state.
Wait a minute. More ATP is used? But the light reactions create the same amount of ATP as NADPH. But from what you said, the Calvin cycle uses approximately 9 ATP and only 6 NADPH. What gives?
Let me see. Quite being on the spot in front of all my viewers. Well, remember when we described the light reactions, we had photosystem 2 and photosystem 1, and the electrons move almost in a line from PS2 to PS1. We call that linear electrons. However, electrons sometimes, when in need of ATP, for example when, as you mentioned, 9 ATP is produced to the 6 NADPH. To make up for that, the cell uses something we'll call cyclic electron flow, in which an electron flows from PS1 around through a separate chain of proteins back into the electron transport chain between PS2 and PS1. There, a proton gradient is established, and by chemiosmosis, ATP is made, forming more ATP than the ADPH.
Professor, I think I'm starting to get it. But is this the only way carbon is fixed into plants to make food?
No. You see, young David, what we've been exploring is what we call C3 fixation, where the initial product is a 3-carbon compound. Remember I mentioned the fixing of carbon dioxide to Ruby P forms a 6-carbon compound. That's short-lived and quickly splits into 2 3-carbon compounds. Another alternate method of carbon fixation is in C4 plants, in which the carbon dioxide is affixed to a 3-carbon compound by PEP carboxylase. It's a mouthful. But that forms a 4-carbon compound, which is what gets the name C4.
So this alternate method of carbon fixation, does it take place in the same kind of cell?
Yeah, well, no, but in C3 plants it all occurs in the same, plant cell, but, well, when in the C4 plants, there's a spatial or spatial, as some call it, separation of the carbon fixation part and the Calvin cycle itself. And you see, young David, in C4 plants, carbon is fixated in the process I just described in mesophyll cells, which are on the exterior of the plant. And then it's stored as a 4-carbon compound, and then enters a bundle sheath cell, where the CO2 is released from that 4-carbon compound and enters the Calvin cycle to enter the same events as in a C3 cell. But the important distinction is the spatial separation between the mesophyll cell, where carbon fixation occurs, and the bundle sheath shell, Sally sells seashells on the seashore, where the bundle sheath cell, where Calvin cycle occurs.
Where the Calvin cycle occurs.
The Calvin cycle occurs.
Yeah. That is nifty. But what's the advantage of doing C4 carbon fixation?
Are you reading your book?
No. You see, young David, the enzyme Rubisco has a high affinity for O2 rather than CO2. And when this happens, a phenomenon called photorespiration occurs, where the Calvin cycle is essentially inhibited because O2 binds rather than CO2. However, C4 plants are able to avoid this by having the C4 process in the mesophyll cell. Essentially, it concentrates the CO2 and prevents O2 from ever reaching the Rubisco, thus creating a very efficient mode of carbon fixation.
Idiot.
Are these two processes the only two that exist? What about, say, in a desert plant that has trouble keeping their stomata open because of the heat?
Well, young David, you see, that problem is solved by crustacean acid metabolism plants, or CAM, as we call them in the business. These plants, they open their stomata only at night, and they absorb CO2 and store it in organic compounds for the next day. When the sun comes up, they close their stomata to conserve water, and CO2 is released from the organic acids, and the Calvin cycle ensues.
So a plant cell can make its own food. Who knew?
I did.
And now you do too.
Stay alive and see the peace in every eye.